Okay, welcome to Mechanisms of Cancer. Today we're going to be talking about the anaphase promoting complex, or the end of mitosis, which is the same thing. So let's go ahead and get some bearings as to where we are. So what we're looking at is the cell, if you remember in prophase, metaphase, where all the chromatids are lined up in the center of the cell. Now, they can't actually separate, and the cell is, believe it or not, stuck in basically metaphase. So right now the M-cyclin CDK complex is active. It is disabling the lamins, which are disabling the nuclear envelope. It is activating the microtubule organization centers. It is making sure that DNA is condensed. So this cell right now is stuck. It can't gene express. It can't basically do anything to react. So it's in a state of stuckedness, which is an official biology term. Definitely. You might want to look that up at Wikipedia. Anyway, so what we have to figure out is what the cell is going to adapt is a way to, what the cell is going to generate is a way to destroy MPF, and that is through the action of a ubiquitolator named APC. So the APC, or anaphase promoting complex, is also sometimes called the machine of the spindle checkpoint mechanism. So there are different conformations that microtubules can be attached to sister chromatids. You can have double attachment, one on each side. You can have attachment from both from one side. You can have single attachment, and then you can have double attachment on the same side. So imagine your chromatids here. You have one microtubule going from here, and you have the same chromatid being attracted from the other side. All of these will basically produce a signal. That signal, and if you want to look it up, you can look up the MAD2 signal, is basically going to only stop being produced by the kinetochore when there is even stability, when there's stability in the kinetochores. So that's when the microtubules are hitting the kinetochores from opposite side and basically pulling on it equally. So once that signal is diminished, that's when the APC starts to become activated. So that'll happen in metaphase when all the chromatids are lined up, sister chromatids are lined up in the center of the cell. They've been hit with microtubules from both sides. So there are really two actions of APC that we have to focus on. When the microtubules have attached, when that MAD2 signal has gone through, CDC20 is activated, and CDC20 helps to activate the anaphase promoting complex. Lots of experiments have been done. Well, we don't need to go into them. Anyway, so the APC CDC20 complex is going to first ubiquitolate something called securin. Now, securin is the inhibitor of a protein called separase. Separase will destroy the linkage between sister chromatids. So attached to the sister chromatids are kinetochores, and those kinetochores have little motors on them. And as the kinetochores are active, so they're actively trying to go away from each other on the microtubule. What's holding them together is a protein called cohesin. Now, cohesin. Now, separase will destroy those proteins called cohesins if it wasn't normally inhibited by a protein named securin. So CDC20 in combination with APC adds these little ubiquitin molecules to securin. And so basically what that is, you attach, I think, about 7 of those molecules, and then another enzyme, a proteasome, will come in and destroy the securin. Once the securin is destroyed, the separase is free to take those sister chromatids, destroy the linkage between them, then those kinetochores will pull the sister chromatids apart. In addition to destroying cohesin, the APC is going to destroy the M-cyclin CDK complex, or MPF. It's going to do that by the same mechanism. It's going to ubiquitolate them. So basically, it marks them for destruction. APC doesn't actually destroy them, but it marks the MPF for destruction. The MPF will be marked for destruction. A proteasome, which I sometimes draw as the Death Star, will come in and destroy that protein. And that will be the end of MPF, which means that the cell is no longer being held by the actions of MPF. So the nuclear envelope will be free to reform, the DNA will decondense, the microtubules will kind of retreat and stop going crazy, and the cell will exit mitosis. So the APC destroys MPF, which kind of reverses its effects. That means that the cell exits. And we'll take a look at this video here just to, no, I don't know. See what's going on. So this is the stabilized conformation with the kinetochore being symbolized by those pink proteins. Now those are sister chromatids that just got pulled apart. They're exact copies of each other. And there's basically your beast. Okay, so that is essentially the end of the cell cycle. Congratulations. We're going to talk a little bit more about a few more signaling things, but that's it. Now, the important thing with all of the stuff that I've taught you is you have to think of two things. You have to think of how these small mechanisms could be altered to drive a cell forward through the cell cycle at all times. Now, cancer is not just a disease of a cell that is able to go through the cell cycle at all times, but that's one of its major components. You also should think about ways that you could disable proteins to arrest the cell and cell cycle. Basically, the targets of cell cycle protein, basically the targets of chemotherapies or anti-cancer agents. So, almost everything that you study in the cancer project is going to link into one of the proteins that we've just seen. I picked those proteins because they're very, very popular in cancer pathways. So you're going to see an RBE2F or a MIC or a RAS or a RAF or a CDC20 or BCR-ABLs, all of these proteins are going to link into one of the proteins that we've seen. So again, when you're doing your cancer presentations, especially when you're thinking mechanistically, you're going to want to look for the names of some of the proteins that we've discussed in the mechanisms of your cancer therapies and therefore the mechanisms of the cancer they're attacking. So good luck on that. Again, I really wish you well on your cancer projects. I'm looking forward to seeing them.